and welcome to the University of Idaho's 2016 Pacific Northwest Clean Water Association Student Design Presentation. My name is Taylor Romenesko. I am a graduate student in the Civil Engineering Department here at University of Idaho.
My name is Bryant. I'm also a civil engineering student, but I'm a junior here at the U of I.
And my name is Karina Eyre, and I'm also a junior studying civil engineering at the University of Idaho.
Currently, the Moscow Water Resource Recovery Facility performs biological nutrient removal on an average of 2 million gallons per day. The wastewater comes into the headworks where it goes through screening and then grit removal. The primary clarifier that is seen in the bottom left of this picture is not currently used by the system. That is because the biological nutrient removal requires all of the influent carbon. Therefore, we do not want to remove it in the primary clarifier. For that reason, the wastewater goes from the grit removal through the Archimedes pumps into a modified anaerobic, anoxic aerobic, or A2R process, which is a form of enhanced biological phosphorus removal, or EBPR. After the EBPR process, the wastewater goes into the secondary clarifiers, where sludge is settled, dewatered, composted, and then land applied as a Class A biosolid. The liquid effluent from the secondary clarifiers is dosed with alum to precipitate the remaining phosphorus, and then filtered through an upflow sand filtration. The effluent is then re-aerated, chlorine disinfected, and dechlorinated and discharged into Paradise Creek. Some of the water is also used as reclaimed water for the University of Idaho. Our team was tasked at looking at the benefits of the addition of a fermenter to the Moscow Water Resource Recovery Facility. Specifically, we wanted to look at the fermenter design that produced the maximum amount of volatile fatty acids, also known as VFAs. These VFAs have many potential benefits that include enhanced biological phosphorus removal process stability and resource recovery such as struvite or bioplastic production. Currently, the Moscow Water Resource Recovery Facility is in compliance with their National Pollution Discharge Elimination System, or NPDES, permit. However, they have had some process upsets in the past, and these process upsets have stopped biological phosphorus removal. These upsets were due to an increase in the amount of nitrate being pumped back to the anaerobic zone through the RAS, which turned it anoxic and not anaerobic. In 2012, in December of 2012, a remedy was installed. The remedy was a nitrate probe in the return activated sludge. This probe would then control the mixed liquor return from the end of the aerobic phase to the anoxic phase, which promoted denitrification. This process upset shows the sensitivity of the enhanced biological phosphorus removal process and the potential benefits of adding a fermenter. Another benefit of adding the fermenter would be a decrease in the alum use and the chemical sludge that is produced. Our team looked at a couple specific project objectives, including forecasting influent flow and pollutant loads for the city of Moscow through the design year of 2041, which corresponds to a design life of 25 years. We were tasked with investigating 3 fermenter alternatives and their corresponding VFA production. At the end, we selected a final fermenter design to analyze the process impacts of that fermenter.
The first step in our investigation was to determine the population for our design year of 2041. Data was obtained from a plan done by the City of Moscow, the 2012 Comprehensive Water Service Plan, and it included a population estimate through the year 2060 using a 1.42% annual growth rate. This turned out to be 35,531 people in our design year of 2041, which is an increase of over 10,000 compared to present day. We obtained flow data from the Moscow Water Resource Recovery Facility staff for the years 2002 to 2015, and we used this data along with the population data that we forecasted to come up with some design flows. The design flow rate that we used for most of our sizing was 2.93 million gallons per day, with a peak hourly flow of 9.37 mgd. Wastewater characteristics were obtained through testing done by the University of Idaho Environmental Research Group, as well as data supplemented that was provided by the staff at the Resource Recovery Facility. Our influent wastewater is pretty comparable to medium strength wastewater. Data was obtained from Metcalf and Eddy. We have 235 milligrams per liter of influent TSS, total COD of 556 milligrams per liter, a readily biodegradable COD of 111 milligrams per liter, and a total influent phosphorus of 3.24 milligrams per liter.
So one of the most important aspects of designing a fermenter is to understand the fermentation process. So essentially what fermentation does is it takes the influent carbon in the wastewater and transforms it into a more useful form of carbon for the microorganisms that one has in their culture. So pictured here we have influent carbohydrates, proteins, and lipids, which represent the influent carbon to the wastewater treatment plant. In the fermenter, it will go through hydrolysis, which will break down these forms of carbon into simple molecules. These simple molecules are then reconstructed through acetogenesis and acetogenesis to create volatile fatty acids. Primarily in wastewater, we find propionate and acetate being formed as VFAs. However, butyrate can also form. In terms of fermentation operations, an SRT of 2 to 6 days is typically recommended. This will vary with temperature, so fermentation will proceed faster in the summer months. So that is an operational parameter that one must consider. The SRT is pretty important because if the process is allowed to go, if the SRT is extended beyond that, the process will proceed to methanogenesis, in which all the volatile fatty acids are destroyed. So it would make the fermenter that we installed obsolete if it was not actually producing volatile fatty acids, and it also produces methane gas, which is a greenhouse gas and should be avoided. For our VFA production yields, we are assuming that we are using volatile suspended solids and readily biodegradable COD as the basis for that VFA production estimate. And we are also operating under the assumption of complete transformation of the readily biodegradable COD into VFAs. So how VFAs actually aid in biological phosphorus removal is pictured in this diagram. So these are known as PAOs, or phosphorus accumulating organisms. And so essentially what happens is that in the anaerobic zone, the VFAs being present encourages these cells to release all of their phosphorus into solution. They then uptake the VFAs into their cell and use that as part of their metabolism to survive, grow, and do all of their great things. Then, downstream in the aerobic zone, long after the VFAs have been depleted, they will uptake the phosphorus back into their cell that they have released into solution, and as part of that will also take up some of the additional P that came into the wastewater treatment facility as phosphorus. This is shown also here in a time scale. So the red line indicates VFAs, which are depleted very quickly from solution, and then you can also see with the blue line as the phosphorus is released into solution. And then downstream in the aerobic process, it plummets as it's being taken back up into their cells. And something else I want to point out, so the phosphorus level does drop after the second uptake. So volatile fatty acids can also be used for resource recovery purposes, one of which is struvite, which is pictured right over here. It is a crystalline form, also known as magnesium ammonium phosphate. Most commonly it is associated with maintenance issues at a treatment facility because it will precipitate out of solution, primarily at low pressure zones within the system, and causes lots of problems in terms of maintenance with pumps and piping. However, if one purposely produces struvite, it can reduce some of these maintenance headaches while also producing a fertilizer. Struvite is very rich in phosphorus, so it can be land applied and therefore repurpose the phosphorus that's accumulated at the treatment plant towards something that's a little bit more useful. And the way that you get struvite production in terms of for removing it from solution is that you use the waste activated sludge line and you pulse it with VFAs to encourage those PAOs to release their phosphorus once again. And we can get about 40 to 60% removal of phosphorus from that waste stream. Also, bioplastics are another option for resource recovery. They are polyhydroxyalkanoates, also known as PHA. Currently, it is a product that is under investigation with the Environmental Engineering Research Team at the University of Idaho. It is not currently completely feasible on a large scale, but it could be something that could be added in the future. So because we have these VFAs and we have a couple of different purposes that we could use them for, we decided to create a hierarchy of where they would go and where they're the most important. So as part of the EBPR process, something that has to occur before that can even take place is the reduction of nitrate. So VFAs will be preferentially used for the reduction of nitrate in the anaerobic zone. After that occurs, they can then be used for EBPR. Any VFAs that we have remaining after that point can be used for either struvite production or PHA. So in designing our fermenter, we had a couple of parameters that we used for evaluating all of our designs. So we will be using no thickeners. We will be operating at an SRT of 5 days. We will reintegrate the primary clarifier into the system, and we will use the same primary clarifier for all designs. Also, our VFA production will be based on theoretical yields from stoichiometric conversions and also through biowind modeling. And the design alternatives that we have selected to investigate are an inline fermenter, side stream fermenter, and an activated primary clarifier.
The first fermenter that we looked at was an inline fermenter. An inline fermenter ferments the entire waste stream, both the liquids and the solids. It is fed primary solids back from the primary clarifier to sustain a stable microbial population within the inline fermenter. There is also a wasting line of the primary solids downstream to the EBPR process. This allows operational control of the solids retention time. Evaluating the inline fermenter had its advantages and disadvantages. A couple of the main advantages were that it doesn't ferment the entire waste stream, therefore it produces more VFAs because there's more carbon available in the entire waste stream. Also, the hydraulic retention time and the solids retention time are independent of each other because of that wasting. That gives operations staff more control over the fermenter to produce the maximum amount of VFAs possible during the summer and winter months. A couple of disadvantages of this fermenter is that it does require a large volume. That is due to it processing the entire flow stream, so it has to be bigger to accommodate that much flow. There's also been research that suggests a slightly larger phosphorus release in inline fermenters than other fermenters.
The second design alternative that we investigated was a side stream fermenter. So under this configuration, primary solids from the primary clarifier are directed to a side stream fermenter. That once they're in the fermenter, they will remain there for the 5 days solid retention time before being directed into the EBPR process. A couple of notes on this particular configuration. A lot of times in a side stream configuration, there will be a wasting line. However, because Moscow already handles all of the influent solids, this is not going to be necessary because we do not have to redirect solids to solids treatment. So therefore, in this configuration, HRT and SRT are going to be equal. So a couple of advantages and disadvantages. In terms of some advantages, because you're only fermenting primary solids, you do not have to have nearly the size of a tank that you would with an inline fermenter. So that can be advantageous if you don't have a lot of space, or if you do not want to have the capital costs of installing a larger tank. Also, it is easily incorporated into other side stream treatment processes, especially struvite. And this is because the VFA effluent is a much higher concentration out of that side stream fermenter because, again, you're only fermenting the primary solids. It's not diluted with that excess water that is seen in the inline fermenter. However, there are quite a few disadvantages. They accumulate fibrous material and scum very frequently. 2 side stream fermenters were recently installed in the Pacific Northwest in Boise and Meridian, and both have had enormous scum issues, sometimes accumulating 2 to 3 feet of scum in the bottom of these tanks, which is a maintenance nightmare and means that it has to be taken down and offline very frequently. They also have frequent odor issues, again because they're fermenting primary solids. It's at a much higher concentration, so more odor issues are realized. And probably the greatest disadvantage of the side stream fermenter is that you're only fermenting approximately 30% of the total COD and 60% of the volatile solids. And this is because you're only targeting what you can remove from your primary clarifier, and So your VFA production is limited when you could be touching so much more of the carbon that's coming into the plant.
The last fermentation alternative that we investigated was an activated primary clarifier. This is probably the most simplest, and basically it's just a sludge blanket that's maintained in the activated primary clarifier. Primary solids are then pumped back into the main waste stream where they are mixed to elutrate the VFAs. They go back into the activated primary clarifier, and the supernatant is discharged into the EVPR process. A few advantages of activated primary clarifiers are they're very cost effective and they can be added to any system that has a primary clarifier. They're most commonly seen in treatment plants that have extra primary clarifiers and are just looking to attempt to increase their VFA concentrations. But there are quite a few disadvantages, most stemming from the thick sludge blanket that must be maintained. This can lead to methanogenesis, and the sludge blanket can also go septic. And because of this, it has to be emptied out and cleaned pretty regularly in order to prevent these from occurring. The sludge blanket's about 5 feet usually, and so we require very deep clarifiers in order to do this. Otherwise, you'll have a very low HRT. APCs have had really mixed successes. Sometimes they work, but a lot of times they don't, and they're just generally very unreliable. And empirical data does suggest that they have lower VFA yields than comparable alternatives. Because of this, we decided not to investigate APCs any further due to their inherent unreliability. So the next step was to compare side stream and inline VFA yield production in pounds per day. On the left in gold, you can see the required VFAs, and for side stream, we calculated stoichiometrically the yield, and we put it in BioIn, and we got about the same number. However, it was not enough to achieve EVPR. On the inline fermenter, we have about a 30% difference between our theoretical yield and our BioIn yield. However, in both cases, this is still enough to achieve EVPR with excess VFAs. Based on this, our recommendation is the construction of an inline fermenter. It has the highest VFA yields, and there are plenty of excess VFAs that can be used for resource recovery, such as struvite or possibly bioplastics further on down the road once the technology has been further developed.
So for the inline fermenter, if we were to implement this, it would look similar to the picture on the right. It would be just under 500,000 gallons at 488,000 gallons. Our design team chose a depth of 20 feet, and we could use either a circular basin or a rectangular basin. Our team chose a circular basin because the ease of mixing that's involved. The circular diameter would be 50 feet, and this volume would allow for a design hydraulic retention time of 4 hours and a design solids retention time of 5 hours, which could be varied. The current system has room right now for an inline fermenter exactly where we would want it, in between the headworks and the primary clarifier. The space is tight, but it is possible to be able to fit it into there with pumping and piping considerations. The first step that would have to be changed in the process if the implementation of an inline fermenter was realized, the first would be the modification of a primary clarifier. The current primary clarifier is a 55-foot diameter primary clarifier that has adequate surface overflow rate of 1,000 gallons per day per foot squared. However, it is not deep enough to provide the hydraulic retention time that is needed to provide optimal performance for maximum VFA yields. Therefore, we recommend to increase the depth from 9.5 feet to 12 feet deep.
A couple of other process impacts that would need to be considered. So in order for a fermenter to act at optimal conditions, it needs to be completely mixed. Pumps would be installed to ensure that this occurs. One advantage of an inline fermenter is that because you're mixing something that is much more dilute, the energy that must be put into an individual unit is much less, even though you are mixing a larger volume. Another impact that is possibly foreseen is chemical phosphorus removal changes. So currently they are using alum to coagulate that last bit of phosphorus and prior to that upflow sand filtration. However, if the EVPR process is stabilized, it is possible that alum use could be reduced, if not completely eliminated. This would reduce chemical costs for the plant, as well as reduce the amount of chemical sludge produced, which is more environmentally sound. Another process impact is struvite production. So if the additional VFAs that are being produced if they wanted to repurpose those, then we would recommend putting in a patented struvite process, either the multi-WAS from multi-ground harvest or WAS drip process or something similar. It's diagrammed over on the right. So essentially what occurs is VFAs from the inline fermenter would be fed to a WAS release tank, which has a portion of the waste-activated sludge strip. What occurs in that tank is the secondary release of phosphorus into solution. At this point, additional chemicals would be added to encourage that precipitation of the struvite in the struvite reactor. A couple of things, so this would be a substantial facility construction, so it's not a small feat to insert this process, but could have a couple of large advantages. The release tank that we've sized will have hydraulic retention time of anywhere from 5 to 15 hours. 15 hours is typically recommended, but we determined 5 should be sufficient for that phosphorus release. Also, in terms of flow diversion, it will only divert about 10% of the design flow from the mainstream treatment process, which should not affect anything else within the plant. Design flow rates can vary that much within anyway, so it should be fine. And then probably one of the greatest things about struvite, in addition to it repurposing the phosphorus, is it could serve as a potential revenue source for the treatment facility to offset operational costs because you can't sell struvite. So in conclusion, we selected an inline fermenter as our design for if a fermenter was to be added to the Moscow wastewater treatment facility. And we selected this because it has enough VFAs to stabilize their EVPR process and has remaining VFAs that could be used for struvite production and possibly bioplastic production as the processes are further refined. Process impacts will all be upstream of the EVPR process, and maximization of existing infrastructure will be realized by modifying that existing primary clarifier.
So we would like to give thanks to Dr. Eric Coates and Dr. Patricia Kohlberg, as well as the Moscow Water Resource Recovery Facility staff and the University of Idaho Environmental Engineering Research Group. This concludes our presentation. Thank you for watching, and we look forward to entertaining your questions at a later time.